Hi everyone, my name is Dea Gogishvili and I'm happy to share with you the findings of my latest research conducted at Vrije University, Amsterdam. In this study, I explored CSF biomarkers and their ability to predict the rate of cognitive decline in dementia. The results of this study have been published and are available online. You can simply scan the QR code provided below and dive into the details. Dementia is a clinical syndrome characterized by cognitive impairment. It poses a significant hurdle to the daily lives of countless individuals worldwide. Although remarkable progress has been made in the field of biofluid biomarkers, providing enhanced diagnostic capabilities for various neurodegenerative diseases, the main question after the diagnosis remains quite difficult to answer. The key question for patients with dementia is the uncertainty surrounding their future and how long they can maintain their independence. The rate at which dementia progresses is highly variable, even within the specific types of dementia. Consequently, the absence of reliable fluid biomarkers to predict prognosis remains a challenge. Cerebrospinal fluid, or CSF, reflects the biological state of the brain and can provide valuable insights into the progression of dementia. The objective of this study was to explore the potential of CSF proteins as biomarkers for predicting the speed of cognitive decline, while also investigating shared underlying mechanisms that can contribute to the cognitive decline rates. In this project, we used data obtained from highly sensitive and specific multiplex immunoassay-based proteomics technology. We defined fast and slow decliners for the classification model based on their mini-mental state examination scores over time and the survival follow-up. To create a clear group contrast, a group of ambiguous patients in the slow decliner group with a survival follow-up with less than 5 years were excluded from the group comparison and machine learning classification analysis. To identify which of those proteins contribute most to differentiating fast and slow decliners, a random forest classification model was trained on protein relative abundance values in CSF. The prediction task in our case was the progression group. We trained 4 different random forest classifiers. The first one was the Olink plus age model, which was trained on all protein measurements and included age as a feature. The Olink model, which was only trained on all protein measurements. The age model, which incorporated only one feature, age, and the random model, which was trained on all protein measurements with shuffled labels. Using protein measurements as features results in a better classification model than only using age. And incorporating age as a feature results in a similar list of top biomarkers. All 3 models clearly outperformed the random model, which was trained on shuffled labels. Here you can see the feature importance values and the correlation direction of the top 20 biomarkers. The analysis revealed that the top 3 proteins ranked by their importance were CLEC1B, TNFRSF4, and TGF-beta1. In order to better interpret the model performance, Shapley additive explanations, or SHAP values, were calculated. For local interpretability, we demonstrated 2 correctly classified patients from the test data to explain which biomarkers contributed to these predictions. SHAP analysis revealed several biomarkers that consistently demonstrated predictive value across multiple patients. For instance, the first patient showed elevated levels of TGF-beta1, which was identified as a significant predictor for a slower cognitive decline. In another case, CLEC1B and TNFRS4 and other biomarkers highlighted in red were found to be important contributors to the patient's predicted fast progression. Importantly, we observed a complex pattern of biomarker interactions for each patient, indicating that accurate prediction may require a multi-biomarker approach. In order to gain insights into the biological mechanisms that might be differentially affected in the fast and slow progressors, we conducted the pathway enrichment analysis. To adjust for age and sex, we assessed which protein expression values differed between the progression groups combined with feature importance results. Most of the proteins with significant differences between the fast and slow decliners showed negative effects, indicating that lower protein concentrations are associated with faster progression. Biological pathway analysis showed the enrichment for GO terms and KEGG pathways associated with cell adhesion, cell signaling, and immune response pathways. As a final step, we were interested in the ability of individual biomarkers from our panel to predict cognitive decline. Here we took the reversed approach and grouped all dementia patients based on their expression levels of our biomarker leads. For each protein, we selected patients with the lowest and the highest quartile expression values and assessed the association with cognitive decline. 11 out of 20 top biomarkers showed a significant difference. To summarize, we identified several candidate CSF protein leads that might carry prognostic value and can potentially help predict the speed of the disease progression in dementia patients. Together, these results suggest that a CSF biomarker panel following future validations can potentially offer useful prognostic information. I would like to thank our co-authors, our funding sources, and of course you for your attention.